Okay, welcome back. So here we are finishing up this last portion of the cell division, mitosis, cancer, meiosis lecture. And so here's a diagram showing meiosis 1. So remember, in meiosis 1, the goal will be to go from diploid set of genetic information to haploid. So at the end of meiosis 1, we want to have haploid cells that have a single chromosome of each kind. So just like we had in mitosis, prophase is mostly concerned with condensing the chromosomes. Now here's an additional feature. Not only do we need to condense the chromosomes, but the pairs, the homologous pairs, have to find each other. And so that's what you can see right here. I guess denoting the pairs, the mother, one's from your father and from your mother, with colors. So we have the red one of each kind and the blue one of each kind, and they have to find each other as a pair. There's something interesting that happens sometimes in meiosis 1. As the pairs are finding each other, they just keep hugging and dancing around one another, and sometimes they exchange pieces. That's called crossing over. And you can see that this has happened here, because now take a look at the bigger one of these two pairs of homologous chromosomes. So the blue one all of a sudden has a red cap, and here another red cap, and the red one has a blue cap. So that means these guys have exchanged even pieces as they were finding each other in prophase 1. So once they are lined up here as pairs in this equatorial plane in the middle of the cell, then it's time for them to say goodbye, and they will be pulled to opposite poles of the cell in anaphase. And moving on down here, then as a result of meiosis 1 in telophase, we're going to have two new cells, and both of them are haploid. Now take a look. Both of the chromosomes that are shown here, they still have two sister chromatids each, okay? And that's what we're going to take care of in meiosis 2. Meiosis 2 is basically a mitotic division. Sorry, did I say meiosis? Meiosis 2 is a basically mitotic division. So in prophase 2, we have here the chromosomes are still kind of condensed, but you can recondense them a little bit. Then they have to line up single file in metaphase 2, right here, single file now. And in anaphase 2, we're going to separate the sister chromatids from one another so that each chromosome in the end has only a single sister chromatid. And so here at the end, when we are dividing up the cells and have two new cells each, so 4 total, they're haploid cells. And each chromosome has only a single sister chromatid. So that's the result right here. So this is generic, not considering whether we're doing spermatogenesis or oogenesis. We'll take a look at it here. Next slide is spermatogenesis. So we are producing sperm cells. They are equally viable, and it might be a little bit easier to look over on this side here of the screen. So we're starting out with the stem cells. Spermatogonium is a stem cell, and when it divides, it creates another sample of itself, another stem cell, and then the other one is committed to undergo the steps of meiosis. So we're going to do meiosis 1 here first, and note how we go from 2N to 1N. These are now haploid cells. So now they're called secondary spermatocytes. So the primary spermatocyte is still 2N, and then here we have haploid cells after meiosis 1 has concluded. Then they're called secondary spermatocytes. Then they have to undergo meiosis 2, and so that creates then 4 haploid cells at the end. They still need to mature. They go from early to late spermatids. They need to grow a flagellum and just do some other maturation steps. In the end, we have spermatozoa, or otherwise known as sperm. And over here, same thing. Okay, so that's spermatogenesis, creating spermatocytes. And here, the events of oogenesis, a little bit different. Now, we still will generate 4 haploid cells. Let's take a look down here. 4 haploid cells, but you can tell that they're not all the same. 3 of them are tiny and 1 is huge. So the events of oogenesis are as follows. You're starting also out with a stem cell, the oogonium. And that one is still diploid, and then we're going to have to undergo meiosis 1, and that will create a secondary oocyte right here. And basically we are unevenly dividing up the cytoplasm of the cell. So one cell, one of the resulting cells here in meiosis 1, will get everything, and the other one basically gets no cytoplasm. It's called a polar body, and it contains only the genetic information, so the other set of genetic information. This little polar body will go on and finish off meiosis 2 and create 2 haploid polar bodies, but they will just disappear. They're just going to die. And this secondary oocyte, the one that got all the cytoplasm, that will then grow in a Graafian follicle, and pretty much it all coincides with ovulation, the ability to be fertilized, and the steps of meiosis too. They pretty much all coincide. So as the egg is able to be fertilized, you can see this little sperm right here is fertilizing the cell. That's when this oocyte here, the secondary oocyte, will finish the final steps of meiosis 2. We're creating one more polar body. Just the extra genetic information that is not needed will be placed into another polar body. And the ovum here that's ready to be fertilized gets all the cytoplasm as one very large cell that then is ready to be fertilized. So kind of the steps are still the same, but the result is a little bit different in oogenesis. You're creating 3 polar bodies that are haploid and 1 ovum that can be fertilized. So here's a slide that's showing you the event of crossing over. I mentioned that earlier. This happens during prophase 1 of meiosis. And what happens is that we have the ability to exchange equal pieces here. So as the pairs of homologous chromosomes find each other to line up in pairs for meiosis 1, they sometimes decide to exchange equal pieces. Now, you should ask yourself, why would they want to do this? It's actually pretty risky, because any mistake would lead to chromosomes that don't work, that are mutated and may have missing information or extra information. So it's pretty risky, but nevertheless, nature does it pretty frequently to ensure more variation. So we already have the idea here of independent assortment. Let me just switch over to the next slide real quick. So independent assortment means that the pairs of homologous chromosomes can line up either way. There's always 2 choices. It's basically a binary problem. It's like flipping a coin. So each pair could line up the blue one on top and the red one on the bottom, or the red one on top and the blue one on the bottom. So for each pair, you're going to have that option. And so if you add up these options for 23 pairs, so that's 2 to the 23rd power. That's a lot of different ways you can line up 23 pairs. Now, in order to enhance that and create even more options for different gametes to form, so genetically different gametes, these pairs of homologous chromosomes, they can exchange pieces, which happened here. These 2 exchange equivalent pieces actually twice down here on the bottom too, and that's known as crossing over. So both crossing over and independent assortment are for the purpose of genetic variation. And you can see here they're both crossing over and independent assortment have achieved what you wanted to achieve, which is 4 gametes that are haploid and all are slightly different. They all have a big and a small chromosome, but they all have different variations depending on what you inherited from your maternal and paternal genes. So now that we have gametes, we can do fertilization again, and that will create diploid cells from the haploid cells. So here again, both parents, the gonads have undergone meiosis and have created haploid gametes. Now, in fertilization, these gametes will fuse and make diploid individuals again. If it so happens that an X chromosome carrying sperm from the father fuses with an ovum, then you will have a baby girl, because the 23rd pair is 2 X chromosomes, and that would be a female. And if it so happened that a Y chromosome carrying sperm fuses with the ovum that can only carry an X chromosome, because females only have X chromosomes for the 23rd pair. So if that scenario happens and an X and a Y chromosome fuse together in fertilization, then you're going to have a male, an XY genotype. Sometimes problems can occur during meiosis. The biggest problem would be nondisjunction, failure to separate. That can happen in two places. It could happen during meiosis 1 or during meiosis 2. Either way, it will create the problem that one of the gametes has too many chromosomes and the other one has not enough. So here's nondisjunction in meiosis 1. As the pairs are lining up, they're supposed to separate. This pair fails to separate. They both go into one cell, and this cell here gets none of the big copies of this chromosome. Now, then they undergo meiosis 2, and now you end up having gametes that have N, a haploid set, plus an extra 1 plus 1. And here in this case, we have N-1, we're missing a chromosome. The N-1 situation almost always leads to a non-viable embryo. The N+1 situation can sometimes lead to a viable embryo, as long as it involves a fairly small chromosome or an X chromosome. That is also a possibility that is survivable. Here, briefly, meiosis 2 can also have the problem of nondisjunction. In this case, the two sister chromatids fail to separate, and that leads to the same problem, N-1 or N+1 on the resulting gametes. Now, if the extra chromosome involves a very large chromosome, like 1, 2, 5, any of these big ones, these embryos will not survive. They will just end up being miscarriages. But there are a couple scenarios where this is possible, survivable, and one of those scenarios is when it involves chromosome number 21, which is a tiny one. And if there's a third one, an extra chromosome number 21, and then most of the time these embryos actually survive and are born, and we know this as Down syndrome. So it's trisomy 21, 3 copies of chromosome number 21. Now there's nothing wrong with that third chromosome 21, but it does mess up sort of the balance in the nucleus and lead to problems. As females get older, since the final steps of meiosis occur right around the time of fertilization, it's kind of a bad term, but the eggs get older as we age too. And so basically the chances of nondisjunction occurring are higher as these eggs already have been exposed to potentially environmental damages. So we see a steep increase in Down syndrome babies as the mother gets older. You can see that right here. And our last slide. Okay, so I'm not sure what happened here with my frame, but anyway, let's focus here on our last slide. Here are a couple other scenarios when the monosomy or trisomy involves the sex chromosome, so the 23rd pair. Surprisingly, even though the X chromosome is very large, what happens is when a male has an extra X chromosome, so XXY genotype, that's called Klinefelter Syndrome. Turns out it's actually not a huge problem. Most guys don't even find out they have it. This diagram seems to indicate they look very female, but it's not even the case. This is kind of exaggerated. Most guys find out that they have Klinefelter if they try to have children and then they find out that they're sterile. So the biggest problem for them is usually that they're sterile, but other than that, it doesn't really cause any other like mental problems or any kind of other physical problems. So Let's talk quickly about the other syndrome that's shown here. That's a Turner syndrome. That's a female with only an X chromosome and no other sex chromosome. So the 23rd pair is not a pair, it's just a single X chromosome. This is causes more physical problems, and it leads to also sterile females, and so you can see some of the conditions that are typically seen in this kind of syndrome. A lot of cardiovascular problems here.